Any questions on those, anything like that? So we got about 2 more weeks or so, and then it's the exam. So the exam, we'll cover the endocrine system. I should wrap that up today, or pretty darn close. Then we'll start the male reproductive system, female reproductive system, and probably development. So they're much shorter sections. So about half the next exam is over, endocrine. The other half will be a little bit male reproductive, a little bit female, for a lot of anatomy in those sections, and a little bit on development. I don't have time to lecture and a half maybe on development. We just do the really early development. All right, so let's see, where were we in this thing? We're just kind of surveying down the rest of the endocrine glands, just listing the hormones quickly, kind of their actions that we're worried about. And I think we've gotten to the thyroid. All right, so we've got all the brain, hypothalamus, pineal, pituitary, posterior anterior. I think we did the thymus and parathyroid. So let's look at the thyroid just for a minute. All right. Again, we looked at it in lab. This is its location. That's the larynx, kind of that Adam's apple and make projectile. So just kind of a bilobe gland, little isthmus connecting the 2 bits. That's the micrograph, like we looked at in lab. So again, the distinctive part of the thyroid, it's got these really obvious thyroid follicles in there. Those are made up where they include that layer of simple cuboidal epithelia, which is just, you can see the nuclei out here. And in all of this, that's extracellular material. Now in lab, if I pointed to that, there's several things you could call it. In lab, you just say it's a follicle, I'm good, because they usually put the pointer right in the middle. But you could also say thyroid globulin. You could also say colloid. I think it's labeled both ways in the lab. Colloid is just basically a water protein sort of suspension. That's what it is. Thyroid globulin is a protein, so you could call that a colloid as well. Thyroid globulin is a little more specific. And talk about a little of it in the lab. Let me go down to a slide that has a bit of metabolism. So what you're seeing here, this is one of those follicles. Again, it's just showing you the layer of cuboidal cells, that yellow area, that's non-cellular. And what happens, if you look at this enlargement, these follicular cells, they will pump in iodine against a pretty significant concentration gradient. This represents a blood vessel here. So iodine, which just comes in your diet, is just a mineral, that gets moved in, and the cells of the follicle will actually then incorporate that iodine into one of the amino acids of a protein. This is kind of what they're showing you here. This is a skeletal structure of a protein. You remember a protein has, it's got an amino acid hooked to another amino acid. I'll just call it AA2 for amino acid 3, hooked to another amino acid, and so on. These just represent the amino acids, the little pig spears. Now, if you remember from last semester, the structure of an amino acid is something like this. It had a nitrogen, carbon backbone, but then had this thing I've got circled, the R, it's called an R group. Remember, that's not an atomic symbol. What was the R group? You remember just in general? Could be 1 of 20 different things, right? It's just considered the variable portion. We don't look at it in much detail in Anatomy 1, just pointed out there, that's variable. Those are a few examples you're done with, because we don't look at that much detail. But remember, the R group can be many different things. There are 20 different amino acids, they vary right there. It could be a hydrogen there, it could be a methyl group. These are big rings here. Now, we're not really that interested in the R group, but what you got to keep in mind on this diagram, they're not showing you any of the details of this protein, except these represent the amino acid sort of skeleton, carbon, nitrogen, or nitrogen, carbon backbone. They are showing you one R group. So on one of these amino acids, they've got this 2, and then they've got this ring-like structure. No, the only reason they're kind of mentioning that, this amino acid, they're showing you the R group. The name of it is tyrosine. Tyrosine is just one of the 20 amino acids. Again, it's not a big deal. You got alanine, glycine, tryptophan. This one's just tyrosine. It just has this R group. So they're showing you the R group. So all these cells in the follicle are doing, they're making a protein, like any other protein, but it has a lot of tyrosine amino acids in it. So this protein we're calling thyroglobulin initially, it's just a big chain of amino acids with a lot of these tyrosine amino acids. Then, what the cells of the follicle will do, they'll take that iodine they brought in, that's an I right there for iodine, they'll bring that in and they chemically attach it to the R group of that tyrosine. So what's happened in the follicular cells, they take the amino acid tyrosine, they modify it slightly by adding an iodine to it. Now what can happen, you can add one iodine to the tyrosine, sometimes you will add two, which is kind of the normal procedure. So what you have is thyroglobulin. It's not the hormone yet. It's just a big long chain of amino acids with a bunch of the tyrosine amino acids iodized once or twice. That's what's stored in the follicle. So it's not really the hormone per se. It's a precursor to the hormone. It's just this big long protein with a lot of iodized tyrosine. Now, if you need the hormones from the follicle, which are your T3 and your T4, here's kind of what happens, just in a nutshell. Imagine all these tyrosines are iodized. What the cells of the follicle are going to do when they take in this thyroglobulin, because your metabolism's dropped, you need some thyroxine, they basically take the R group off of one tyrosine and pretty much attach it to the R group of another. So this is now a modified tyrosine, but it's kind of got two of the R groups have been sort of combined. Now here's where it gets kind of weird. If you take two of these tyrosines that have two iodines on them and you combine them, you're going to end up with 4 iodines on this sort of modified R group. But if you take one of these that was iodized once and combine it with one of these that was iodized twice, what are you going to get? 3 iodines. That's the only difference between T3 and T4. It's kind of what they're trying to show you here. There's a nitrogen carbon backbone. There's 1 iodine, 2 iodines, 3 iodines. There may be a 4th right here. It's random chance. Most of the tyrosines are iodized twice. So as a general rule, when you're making tyrosine, most of it ends up being T4. Because if you take one of these tyrosines with 2 iodines, you combine it with another tyrosine with 2 iodines, you're going to have 4 iodines on it, right? That's how you get your T4. There's much less of this tyrosine that was iodized once. So if you combine these two, you get T3, but it's in a much lower concentration simply because there isn't much of this made. It's mostly iodized twice. Now here's my other question. Is it possible to get T2? Yeah, I mean statistically you could. It's possible to take one of these, combine it with another one of those, you got 2 iodines. What does that do? Absolutely nothing. It's kind of a mistake. It'll get recycled. It's not a hormone. But there's some of it made. We just don't think it has any function. So something like 95% of the hormone that's produced is actually in the form of T4. Lesser amount of T3. But they're both released into the circulation. And here's what's kind of weird. In the target cells, all of the target cells have an enzyme that enzyme, when T4 comes into target cells, all the target cells for thyroxine have an enzyme that will convert T4 into T3. They just cut off one of the iodines. So this is always what binds to the receptor. Any of the T3 that comes in, it's already T3, binds to the receptors, causes the change in physiology, raises metabolism. Any T4 that goes into the cell, which is the majority of the thyroid hormone, is converted to T3 anyway, binds to receptors, causes the same effect. So they're really, other than a slight chemical difference, they're the same functionally. When they reach a target cell, everything gets converted into T3 anyway. So I guess you could make the argument, is T4 even a hormone? There are no receptors for it, so really T3. That's why I say they're functionally equivalent in the lab, because they are. Everything that's released, whether it's T3 or T4, gets converted into T3. That's what binds to receptors and causes the effect. But most of what's secreted is actually in the form of T4.
Why does the body even make T4 then?
Why does it?
Yeah, is that like
evolutionary? It's probably enzymatically, you just get most of this made. I suspect it's something along those lines, but I don't know for certain. But when you put the iodine in there and you have the tyrosine in there, the enzyme's just going to add that iodine, and it mostly does it twice. I mean, why, it seems like it makes more sense. Just make the T4 go in there. So I couldn't tell you. It's something to do with, this is the only way the enzymes inefficiently work, perhaps, but yeah. And I think I mentioned in lab, part of the reason we have this huge amount of extracellular storage, and that's unusual in an endocrine gland. Usually if they make excess hormones, they just store it in vesicles within the cell. The thyroid gland stores that huge amount outside of the cell. That's extracellular storage of thyroglobulin, which is a stored form of T3, T4. And that's mainly because of the iodine. Again, amino acids you can make whatever you need. You know, you can convert amino acids one to another. They're in your food all the time. It's easy to get the tyrosine. The iodine is a resource that can be limited. So when you have access to iodine, you know, it's beneficial to make all the thyroglobulin you can in case down the road you don't have access to iodine. So it's really a way to store iodine, not so much the tyrosine that just will get stored. All right, and of course we know most cells have target target receptors. Most target cells have receptors for T3 and T4. You know, so target for thyroxine or T3, T4 is most tissues. There are very few that don't respond. And the other hormone I just mentioned in lab, I won't add much to it. There are a few cells, a little hard to see on this diagram, that are not part of the follicle. Those are those C cells we looked at in lab. A little hard to see on this. So here's the follicle with its colloidal cells. Here's one here, and then they got a mess up here. Looks like the follicle here. You got a few cells that are not part of the wall of the follicle. Those are the C cells or extrafollicular cells. They secrete a different hormone, certainly not as critical a hormone as thyroxine. That's where the calcitonin comes from, which basically controls blood calcium levels. It will lower blood calcium levels if you have excess calcium in the blood. I think I mentioned in the lab, it's really more critical when you're younger. In adults, its function is minimal. We did the parathyroids. I think last time we did the thymus. So let's go really to the last couple. We'll be out of here. Let's do the pancreas. Let's see, pancreas. Again, here it is. And I think I mentioned in the lab, the pancreas is a dual gland. It's an exocrine gland. It's actually part of the digestive system. That's why it has these pancreatic ducts in it. Endocrine glands don't have ducts. The pancreas does, because most of its cells, all these dark ones, those are the exocrine cells or SNR cells. Nothing to do with the endocrine system. They secrete digestive enzymes, bicarbonate. We'll talk about it again when we get to the digestive system. There are some scattered patches of cells. Those are the islets of Langerhans, or pancreatic islands on this diagram. Those are responsible for producing the pancreatic hormones. There's two main ones. They are non-lipid soluble. It's an antagonistic pair. They play a role in controlling blood glucose levels. I think insulin is one of the first hormones we characterized and synthesized early on for hormone treatments. Because, of course, these are necessary for controlling blood sugar. Blood glucose or blood sugar levels, like many internal variables, have to be maintained at a set point. It's like 0.1%. Part of the reason for that, lots of organic molecules can be used as an energy source. You can use lipids, fats. You can burn proteins for energy. But there are a few organ systems, in particular the brain, that can only use glucose as an energy source. They don't have the ability to metabolize fats or proteins or any other carbohydrate for that matter. So if you don't have glucose, your brain shuts off. And that's pretty detrimental for mammals and primates in particular. So you have to have some minimum level of glucose in your blood to keep your brain working. So if it gets too low, you're dead. On the other hand, if glucose levels get too high, it's like giving a pitcher of over-sweet Kool-Aid to a 4-year-old. They start bouncing off the walls, that sort of thing. Well, often that a little bit. High blood sugar tends to overexcite the nervous system. And you can, get a little hyperlocal kid, but then you go into convulsions, and again, a coma, and you're dead. So you got a range where you have to keep this glucose. You have fluctuating like crazy. That's what insulin and glucagon are for, and that's what diabetics don't have. So normally, blood glucose could be around 0.1%. Now, if you go back to this vending machine And you know, you have that crappy quiz, you're depressed, eat 15 candy bars, 15 of them. It's almost all sugar. You digest that, that's glucose in the blood immediately. So if you ate 15, you know, candy bars back there at the vending machine, is your blood sugar going to go up and to this point and dead? No, you don't see students dead over by the vending machine very frequently. It doesn't happen. Why not? When you eat that candy bar, it all goes into your blood. Well, you go up a little bit. But of course then, that glucose in your blood, that directly signals the pancreas, it's humoral stimulation. I think this is the example I use. It'll start releasing insulin. The job of insulin is to lower blood glucose levels. So as soon as glucose starts going up, insulin is released. It targets a number of cells, but in particular the liver. And what it does, it signals the liver to take glucose, pump it active, well, it's an active transport, it's facilitated transport, but pump the glucose from the blood into the liver cells, and that drops your blood sugar levels, your blood glucose levels. It also signals the liver to produce glycogen. You remember that? Glycogen is that polymer glucose. That's how you store your excess carbohydrates. So the blood sugar levels won't go up. You know, never. You don't want them to. If they go up high, you know, it'll kill you. So insulin is going to prevent you from getting up too high in the blood sugar. On the other hand, what if you stop eating? Because, you know, you're depressed. New Year's resolution wasn't to eat 15 candy bars. You're on some kind of diet. So nope, that's it. You go to nothing but a water diet. So you stop eating. Now you start off with 0.1% glucose, but you eat nothing but water. And your body's constantly burning the glucose. So the glucose go down, 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 and then it'll get that low in your life. How long can you go without eating? A day? 2 days? Week? Month? 2 months? Everybody can go months. Of course, it depends on your condition going in, but people have gone months without consuming any food, just water. So you can last a long time. Well, what that means is the blood sugar can't be dropping that low, or you wouldn't last that long. So yeah, the same thing happens. When your blood sugar starts to drop, glucagon is the other hormone from the pancreas. It has an opposite effect. It targets basically the same tissues. It'll target the liver, cause the liver to take all that stored glycogen, chop it up into glucose, release it into the blood. Just your stored carbohydrates will keep your blood sugar levels good for 24 hours. After that, day or two, you don't have any more carbohydrate stores, but glucagon can also initiate, what's that term they use? Gluconeogenesis, it's way too long. But basically, you'll turn other organic molecules, you know, the carbon bits of amino acids, of lipids, you'll start converting those into glucose as well. You have metabolic pathways to do that. And so you'll use any other organic molecule, convert it into glucose, keep those glucose levels high for a long period of time. Now, once you've depleted pretty much everything, you know, lost all your body fat, not much more protein, everything plummets into your toes. But that's glucagon. Will keep those blood sugar levels or blood glucose levels from dropping too far. And of course, that's what diabetics don't have, which was, you know, nearly lethal until, well, this is early onset or insulin-dependent diabetics, until they developed hormone therapy. You know, life expectancy was really, really cruddy because you can't control the blood sugar. You eat too many carbohydrates, boom. You don't eat, boom. But once they had injectable insulin, and particularly if they had a way to electrochemically test your blood sugar, you can mechanically mimic those effects. Of course, in insulin-dependent or type 1 diabetes, the odds of Langerhans gets destroyed. I think it's some kind of autoimmune condition. The immune system really targets and kills those cells, so you don't produce insulin at all, nor glucagon. So those are two main hormones in the pancreas. Again, pretty well known. Now, you don't hear a lot about glucagon. You hear insulin all the time because lots of people use it. You don't usually hear about glucagon because honestly, even if you're diabetic, you don't really have to take glucagon to raise your blood sugar. There's easy ways to do it. You know, eat a piece of bread, eat a candy bar, come right up. There is still glucagon available for emergencies. You know, like go unconscious, you can then apply it and it will bring the blood sugar levels up. But it's not used as frequently as the insulin. That's much more common. All right, now this will be the last endocrine gland we talk about. Then we'll just start the reproductive systems. When we get to reproductive systems, we'll look a little bit more detail at the testes, the ovaries, the reproductive endocrine glands, gonads. Well, let's look a little bit at all right, we already talked about it. You get a bit loud. I'm not going to add too much. But the adrenal gland, kind of like the pituitary, it is a dual gland. You have an outer layer and an inner layer. The inner layer is smaller, the medulla. about 20% of the gland's volume. The remaining 80%, cortex, outer layer. And of course, the medulla is interesting. This is a neuroendocrine gland. Most of the neuroendocrine glands we talk about, pituitary, hypothalamus, pineal, they're all up in the cranium. It's not hidden. So this is basically nervous tissue. I think I mentioned in lab. It just developed from the sympathetic nervous system. It's directly connected to the sympathetic nervous system. Sympathetic nervous system. So the adrenal medulla is stimulated by neural stimulation. Its nerve impulses go down there from the sympathetic nervous system. And when it's stimulated, it releases two hormones. They're functionally similar. Epinephrine, norepinephrine do basically the same thing. They target slightly different glands, have different receptors for them. So they are separate hormones. If you look at the details, they have some slightly different effects. But in general, they basically do the same thing. And what they do for the hormones in the adrenal medulla, just mention something that the fight or flight response does. Because remember, that's what they are. Epinephrine, norepinephrine just reinforce the fight or flight response to improve physical performance. So anything that's going to get more oxygen to your skeletal muscles is a plus. That's things like increasing cardiac output, increasing heart rate, increasing blood vessel diameter or blood flow to the skeletal muscles, restricting it to abdominal organs. You don't need them if you're going to get eaten. And they both do basically the same thing. So they'll target the heart, increase heart rate, increase the pumping ability. So just like the sympathetic nervous system, they just reinforce it. I think I use this as an example somewhere. The sympathetic nervous system, nervous system is short duration. When you activate it, it may only last 30 seconds. But when you dump out these two hormones, it just keeps the fight or flight response going for maybe a couple of minutes, more time to escape whatever it is the situation is. So that is just modified nervous tissue. Have the dark shade area here. Now the cortex. Kind of showing the cortex out here. 2, 3 zones. We looked at it in the lab. They've described as diffuse zones. They are a little hard on some of the sides to tell where one stops and the other begins. It depends on the section, how it's stained, a number of factors. But the adrenal cortex secretes steroids. Again, I told you separating lipid soluble, non-lipid soluble. All the steroids are lipid soluble. They either come from the adrenal cortex or the gonads. So from the adrenal cortex, you may see them called collectively corticosteroids. It just means cortical steroids. So these are all steroid hormones. They come from the adrenal cortex. And you got the 3 zones. 1, 2, 3. Underneath the capsule, again, that's just a connective tissue protective sheet on the outside. You got the zona glomerulosa, fasciculata, and reticularis. Again, there is some mixing of which hormones come from where. But let me go over a few of these. We'll start with the glomerulosa. That's the layer just under the capsule, right on the outer edge. And the cells are kind of arranged as, if you use a little imagination, kind of roundish or oblong, sort of clumps of cells. But the main hormone that comes from this zone I'm going to use aldosterone. Right, that is an example. That's a specific hormone. I will also mention, though, there's a class. I don't like the term much because it's too long to write. But if you see the term mineralocorticoids, it's kind of a mouthful. That's referring to not a specific hormone, but a class of hormones that actually control mineral levels. So that just means, it's a mineral controlling hormone from the cortex, and it's a steroid. There are a number of different mineralocorticoids. Aldosterone is the main one. 95% of the activity of this category of mineralocorticoids is aldosterone. So that's the only one I'm going to talk about. So aldosterone is the main hormone released from the adrenal cortex, primarily the zona glomerulosa. And what does it do? Well, as you can guess by the name, it does a handful of things. But the primary, it controls mineral levels, specifically if aldosterone is released, its main target are cells in the kidney, and this is the first thing it will do. This is probably the most important. Aldosterone does a lot of things. I'm going to kind of rank them in importance. The first thing it does, probably the most important from a survival perspective, is controlling mineral levels of these two ions, potassium, sodium. If this doesn't work, you drop over dead immediately. You know, you remember probably last semester, you looked at resting membrane potentials, action potentials, right? Now, if you remember, I'm not going to review the whole thing, but in general, most cells, if you put electrodes in there and measure the electrical difference across the membrane, from the inside versus the outside, there is an electrical potential on almost every cell. It's called a resting membrane potential. The difference in charge between the inside and the outside has a magnitude of about 70 millivolts. The inside is negative relative to the outside. So if you stuck a couple of electrodes in from a voltmeter, stuck one in here, and stuck one here right next to it, you would measure an electrical difference of about 70 millivolts negative on the inside. Is that ringing a bell? Okay, good. What caused that resting membrane potential? If you remember, if you look at a resting cell, that's any cell, it could be a nerve cell, muscle cell, ovocytes, almost any cell has this resting membrane potential. It's not just unique to muscles and nerves. Most cells have a resting membrane potential. And if you look at the extracellular fluid, there's a relatively large amount of sodium ions outside of cells. And inside of cells, there's very little sodium. It's like, it's at least an 8-fold difference. I'm thinking about 20 millimoles versus 120 on the outside. So there's a huge concentration range. A lot of sodium on the outside, very little on the inside. In a similar manner, potassium concentrations in your cell cytoplasm, they're really, really high. Potassium concentrations out here, which is extracellular, or you can think of it as blood potassium, are very low. Turns out for this resting membrane potential, it's almost all potassium. Potassium is what generates most of that charge that we record as a resting membrane potential. The reason it can do it, remember there's a concentration gradient, right? So which direction does potassium move if it can move by diffusion? Out, right? It's high inside, low outside. It's going to move out. So as the potassium moves out, it's taking positive charges from the inside, moving to the outside. That's what happens. That potassium keeps diffusing out until the inside gets that negative. That is mostly responsible for the resting membrane potential. So that resting membrane potential is dependent on the concentration of potassium outside being fairly low, inside the cells being fairly high. What the kidney controls is the extracellular calcium, or you can think of it as blood calcium. So one of the things aldosterone will do, if your blood potassium level gets too high, these cells cannot generate action potentials anymore, which is really damaging because that means your heart stops and your brain stops. And as a mammal, we're kind of dependent on those two organs. So you cannot let extracellular potassium levels increase very much, or you drop dead, literally. So if potassium starts to increase a little bit, this is another example of humoral stimulation. The adrenal cortex will automatically release aldosterone. It will target the kidney. That makes the kidney dump excess potassium out in the urine. Keep it low. If you didn't have aldosterone or the kidneys don't work, that's what will kill you. I mean, if you increase extracellular potassium just a little bit, you drop dead of a heart attack. In fact, you ever watched potassium is actually fairly safe. It's lots of potassium salts. If you can eat them, it's not going to do much. If you eat potassium salts, you'll get rid of them with your kidneys. It's not a big deal. If you inject them, though, almost immediately, you're dead. If you watch like some of those, police movies where there's a star witness, they were blown up, they're not quite dead, so they're in the hospital. And, they show them a little gurney there, and the heart monitor's going beep, 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 and then the door opens, and all you see is boobs like walking towards the bed. Someone takes a little IV and a syringe, they go, and then they walk away, and you see the heart monitor go beep, beep, beep. Potassium chloride, that's all you have to do. It's not dangerous. It's upstairs, it's later, it's just salt. But if you inject it, it raises blood potassium, no more resting membrane potential, heart stops, nervous system stops, you're dead immediately. Trace 2 potassium chloride, it's already in there. So this is critical. You can't let extracellular potassium go up much, or your heart stops, nervous system stops. So that's the most important. In addition to the potassium, it does affect sodium levels as well, the opposite direction. Aldosterone makes the kidneys pump out, get potassium out of the blood. but it exchanges it for sodium. So as you pump potassium out, the kidney is actually pumping the sodium back into the blood. So the effects of aldosterone on ion levels, on minerals, it will lower blood potassium levels, but it will raise blood sodium levels. So that's why it's called a mineralocorticoid. And that probably its most important role is keeping that potassium where it needs to be, because that's critical. You know what quarter horses are? I'm not a horse person that much, but there was a lineage of quarter horses. I think it was the impressive lineage. There's a stallion impressive. I guess he won lots of things or whatever. And so if you have a horse that makes lots of money because he's pretty, you can breed him and make more money, right? You know, stud feed and whatnot. The horse had a genetic mutation, something on this system here. The ion channels weren't, something's wrong with them. And so all the offspring, not all of them, many of the offspring had issues with this. And what happens in most mammals, if you exercise, blood potassium levels go up a little bit. That actually improves performance a bit. But in this lineage, blood potassium levels will get a little too high. And you know, in minor cases, it causes muscle tremors and you know, things don't work well. In severe cases, every time the horses exercise, run, poof, they die of a heart attack. I mean, so it's really kind of critical to prevent these potassium levels from passing a certain threshold. All right, so that's number one. Number two. The other function of aldosterone is blood pressure control. We'll actually look at this function a lot more. For exam 2, we look at the circulatory system and control of blood pressure. Remember we were looking at hormonal stimulation, neural stimulation, and tropic hormone stimulation. They're not mutually exclusive. So aldosterone, it'll be released due to potassium levels. That's humoral. But there's another circuit that can cause aldosterone to be released, and that one is, I'm going to write it over here because it's kind of long. This is sometimes just called the renin-angiotensin system. I'll go over it again. We get the heart, we get the kidneys. This involves several organ systems. But your kidneys, one of the many things they do is monitor your blood pressure. All right, you got all kinds of blood pressure sensors in the carotid arteries in your neck, around your aorta, and also in your kidney. If blood pressure drops, and again, as we'll see, you know, what's a normal blood pressure for a human? A normal clinical range is what? 120 over 80. All right. What you have to remember is blood pressure, that is the force moving blood through the circulatory system. If it gets too low, you have less force pushing blood through your blood vessels, you'll get less oxygen delivery and nutrient delivery to your tissues. So you need some minimum blood pressure to keep the blood flowing properly and get enough oxygen to the tissues. So there's a minimum you must maintain. If it gets too low, everything starts shutting down and you die. Also, you don't want it to get too high because that overworks the heart, blood vessels tend to rupture, you get heart attacks and strokes. So again, you keep it over a pretty narrow range. But your kidneys can detect a drop in blood pressure. And it could, you know, hemorrhaging. You get an accident, you know, traumatic amputation, all your blood's leaking out. Well, you lose blood volume, and that tends to drop your blood pressure. This is bad. You're going to die if blood pressure gets too low. So in response to low blood pressure, some of the cells in the kidney can detect low blood pressure because there's a huge amount of blood going to the kidneys. About 30% of the blood you pump goes to the kidneys. Those cells will, in response to low blood pressure, will release renin. So this is a chemical signal. What renin does, again, it's kind of an enzyme cascade, it converts a preformed plasma protein that was ultimately made by the liver into this stuff called angiotensin 1. That's what it's called. This forms in the blood in response to renin. Angiotensin 1, when actually that blood is passed through your lungs, there are enzymes in the lungs that convert enzymatically angiotensin 1 to angiotensin 2. Once you have angiotensin 2, a couple of things happen. Angiotensin 2 is a signal. It will target the adrenal cortex and it will cause the release of aldosterone. So this is sort of a tropic stimulation. It's basically is acting like a hormone. So when the kidney releases renin, that leads to the production of angiotensin 2. That's a signal. When it reaches the adrenal gland, it causes the cortex to release aldosterone. And then what does aldosterone do in this case? I've already mentioned this one. It will increase the reabsorption of water and sodium. So it's kind of reabsorbing an isotonic solution from your urine back into your blood. This is a way to boost blood volume and bring blood pressure back up. That's what it's for. So it's similar, but in this case, aldosterone's main role is to reabsorb sodium and water, which is going to boost blood volume, your blood pressure. We'll talk about angiotensin II later as well. That also causes vasoconstriction, which increases blood pressure as well. Let's see, another way, I think that's about it for aldosterone. Those are the two main pathways to get aldosterone to be released. So a drop in blood pressure or a rise in blood potassium levels, either one will directly or indirectly cause aldosterone to be released. So you got humoral stimulation and you got some trophic stimulation. It's not it for aldosterone, that's all we'll cover. Let's look at the next hormone. A little bit. I think that was on the quiz, wasn't it? Cortisol? ACTH. All right, another way to stimulate the adrenal gland, but a different population of cells for the most part, is ACTH, which comes from the anterior pituitary. It's adrenocorticotropic hormone, but it doesn't cause a significant release of aldosterone. Maybe a little, but not a significant amount. Aldosterone is mostly potassium and the renin-angiotensin system. ACTH will cause a different hormone to be released from the adrenal cortex, and that is cortisol, which is sometimes generally referred to as a glucocorticoid. Glucocorticoid, like mineralocorticoid, it's a category. It's not a particular hormone. Glucocorticoids, as you might guess, they're corticosteroids. They affect carbohydrate metabolism, glucose levels. It's one of the things they do. The main glucocorticoid, though, again, makes up like 95% of the activity, is cortisol. So there are some other glucocorticoids you might see in text here and there, but they're of lesser importance, I guess we could say. It's mostly cortisol. And yeah, what does cortisol do? I think I told you, it's kind of like a stress hormone. It does target a number of cells and increase the production or blood levels of glucose, similar to glucagon, but it's for a different purpose. A little bit of glucose is when you're under stress, it can, aid metabolism, energy, that sort of thing. Also, cortisol will lower inflammation. That's often why it's prescribed. Cortisol is one of the more commonly prescribed types of hormones. If you got a rheumatoid arthritis flare up, and your joint doesn't work very well because it's inflamed, they'll just inject with cortisol. I think it stabilizes lysosomal membranes, which tend to reduce inflammation a little bit. And so they use it for allergic reactions. Some of your steroid treatments, I think even for breathing, can be cortisol-based to get inflammation in the airways, for example. Cortisol is really good at reducing inflammation. The downside of that is if you apply it long enough, it does tend to suppress your immune system as well, so you tend to get sick from other things if you overuse the corticosteroids like cortisol. But that is primarily released because of the trophic hormone ACTH from the anterior pituitary. So cortisol, called a stress hormone. And the last one, don't need to talk much about. I'm going to call it testosterone. You might see the term androgens. Androgens is just a general term for male sex hormones, testosterone being the primary. You do get some male sex hormones released from the adrenal cortex. I think I mentioned this in lab. Probably not significant in males because the amount is tiny compared to the amount of testosterone coming from the testes. That's nothing. But in females, this is the main source of androgens from the adrenal cortex. So it may play some roles there, sex drive, libido, that sort of thing. So you get a little bit of male sex hormones from the adrenal cortex. Now, these zones, they are diffused both sort of histologically and sort of functionally. The zona glomerulosa does secrete most of the aldosterone. The zona fasciculata actually secretes cortisol and androgens and testosterone. Same with the zona reticularis. There's a lot of overlap. So you get a little bit of secretion from all of these, all three types. But in general, you get more aldosterone here, much of the cortisol here and the majority of the testosterone or androgens from the zona reticularis. But there is mixing, just like histologically, it's hard to separate one zone from another. There's a little bit of crossover. And I think, other than the ovaries and testes, which we'll cover with reproductive systems, we're kind of done with chapter 17. Now, I think I mentioned earlier, again, this is just a classic endocrine system. As we move through other organ systems, there are going to be other hormones and other endocrine cells that might pop up. You know, like the heart has some hormones it releases, they're not covered in this chapter. The digestive system has a handful of hormones it releases. That's not covered in this chapter. So a few more hormones may pop up later, but that is the bulk of it, and that's all we're going to do for chapter 17. All right, so the next thing. Go ahead and start today. We won't get very far. But the next couple of sections, they're easier. I'm not saying you can take a natural class or something, but not as much to them. We're doing the reproductive systems, which is really heavy on the anatomy. You know, the endocrine system has a lot of figuring out this does this and this affects this. It's much more complicated. Reproductive systems, while there is some physiology going on, you could make it complicated. The way we cover it, it's mostly anatomy. We kind of look at all the parts, and then you get, you know, some brief functions here and there, some secretions and what they do. But it's not as bad. Most people don't have trouble with the male or female reproductive system, although the hormone control of the female reproductive system is pretty complicated, but it's not too bad overall. So we'll start with the male, because it's a little bit easier. I'll start it today. We'll finish it up next time pretty easily, so about a lecture each. But male reproductive system. First of all, kind of separate into two broad areas. The primary reproductive structure, you know, it's the gonads, the testes in a male. Those, of course, produce the male reproductive cell, the sperm, or gametes if you want to be general. Everything else on these diagrams, you could call them accessory reproductive structure. So you got the primary reproductive structure, testes. Everything else is secondary. And all it is, it's a bunch of ducts and glands, and their role is to protect the sperm and deliver it to the female reproductive tract. So it's just going to be a handful of, you know, tubes, ducts, and you have some secretions that are often protected in nature. So eventually we'll basically start at the site of sperm production, some different tubes of the testes, and we'll just name all the parts until we, you know, come out the penile reproduction. So, you know, I've already covered the lab, it should be pretty quick. But before we get there, let's look at the testes for a minute. There are, I think, weird things with testes. Main one being, let's see, where's my diagram? Well, that's the arrow. All right, the testes, like the ovaries, they begin development up in the abdominal cavity. Now, the abdominal cavity is a pretty good place to put organs. But for some reason, in mammals, the testes start up here and then get popping out of the abdominal cavity in this bag of skin we call the scrotum. From an evolutionary sort of perspective, this seems really stupid. I mean, you know, you've heard like natural selection when you look at survival of the fittest, things like that. You've heard those terms, right? Well, in an evolutionary perspective, survival of the fittest, of course, has nothing to do with your strength, how smart you are, or anything else. It's all about reproduction. If you produce no offspring, your fitness is zero. Because really, what happens to your genetic lineage if you have no offspring? It's extinct. You know, that's it. Your genes are gone unless you got some siblings or some other relatives that pass along. You're done. If you have lots of offspring, those genes, you know, increase in, you know, future generations. So it's all about differential reproductive success. That's really what natural selection is. And so when you think of it that way, your testes are critical. If they get damaged and don't work, your fitness is zero, your genes are done. So it seems doubly stupid to take your gonads and hang them out here in a bag of skin where, you know, predators can bite them off, they can get stuck on sticks, all kinds of bad things can happen to them. All your other organs are stuck in your abdominal cavity. Why hang these out there? Why are the testes out there in that scrotum? The answer is temperature. That's not really a good answer, but that is the answer. It's not very satisfying to me. Yeah, for some reason, you know, normal body temperature is, what is it, 98.6 Fahrenheit, something like that. You can't produce viable sperm at 98.6. Usually they don't develop properly, and the flagellum doesn't work, and they can't swim, and so you're basically sterile. They have to be about 4 degrees cooler, so they can't stay up in the abdominal cavity, or they'd stay too warm. In fact, you know, occasionally if a male has testes that don't descend, if they don't basically pull them out of there, that individual will be sterile. So they have to be at slightly cooler temperatures. That's the why. The reason I don't find it very satisfying, all right, birds are the same way. They got ovaries and testes. When those male birds fly over, you see that little scrotum hanging down? It would be very aerodynamic. No, so I don't know how they can get away with it. But in mammals, you know, for some reason, these suckers got to be a little cooler, so you got to get them out of the abdominal cavity. And that's kind of what they're showing you here. Here's the testes getting up here in the abdominal cavity. This little blue thing here, that's a little bit of contractile tissue. It's what they've labeled the gubernaculum there. That actually contracts. You know, later in development, we're talking the 8-month fetus here, so just before birth, that gubernaculum contracts. It's attached to the floor of what's going to be the scrotum, and the testes here, and it just drags it out of the abdominal cavity through that inguinal canal, plop, and then down into the scrotum. And of course, as the testes descend, they drag all kinds of stuff with them. That would be part of the spermatic cord we looked at last. Let me mention one other structure now that comes down with the testes. See this pink thing right here? Pulling down with it. All right, I need to talk about this part a bit. Honestly, it doesn't have a huge role in the reproductive system, but because it's here and it's part of it down the scrotum, I'm going to talk about it now because it will come up several times during the semester. What this pink membrane is showing you, they've got it labeled as this thing here, peritoneum. This is part of what they call the serous membranes. You've got several sets of serous membranes associated with every body cavity. That is, there's a set in the abdominal cavity, there's a set around the heart, there's a set around the lungs. They're all referred to as serous membranes. And they all have at least one function in common. Let me show you what I mean. Let's say we're going to do a cross section through your abdomen. There's your abdominal wall, there's your intestine. Maybe a bit simplified, but imagine you take a section. So you got an abdominal wall, and then you got a tube, the intestine inside. If you look at something's intestine, see, unfortunately for us, we're only looking at dead things. And dead things don't move much. But if you took a fresh thing that's hopefully anesthetized, and you open its abdominal cavity, what would the intestines be doing? Would they just sit there? No, it's not like looking at our dead cats. They're wiggling around like crazy. All your internal organs are moving. You know, your lungs expand, contract, your heart, squishy, squishy, squishy. Intestines are wiggling around everywhere. You know, the only way to get that, jump from one end to the other, 30 feet through a tube, that tube is moving. It's very active. So here's the potential problem you have. You got these intestines wiggling around there, rubbing against each other, rubbing against the inside of your body wall. When you rub two things together, you can get some friction, right? Friction can damage the tissue and cause inflammation. If you get inflammation in there, it's pretty much going to be chronic. It's not like you can stop those organs for a while and let things recover, right? So it's really critical not to generate a lot of friction when all these internal organs are moving around. This is what the serous membranes do. You see, what you have is you have a thin membrane lining the inside of your body cavity. It then folds back on itself and covers the individual organs. That's a serous membrane. They're always a double layer of membranes. One layer covers the cavity, then it literally folds back on itself and covers the internal organs. This space in here, mine is a bit exaggerated, but this is a cavity. It's like, when you open up your cats, we'll do that sometime in a week or two, you can slide your hand in here, right? And there's a space between your body wall and your intestine where you can wiggle around. That's this. I've exaggerated a bit, but that's the cavity between the outer and inner membrane. And it turns out these serous membranes, they secrete a material called, creatively enough, serous fluid. So if you took a fresh specimen, opened it up, poked the abdomen a little bit, stuck your hand in there, rubbed around in here and pulled it out, it's like it feels soapy or oily. It's a lubricant. So these double membranes, they're basically a layer of epithelial and basal membrane. They're fairly delicate, but they secrete serous fluid in here, which is a lubricant. Now all the organs can wiggle around really easy, not a lot of friction, and that's what these serous membranes do. They're all set up basically the same way. Now, to make it a little trickier, in every body cavity, they change the name. Now, they're all called serous membranes around the abdominal cavity, the heart, and the lungs. But, to separate them, the ones around the heart are called pericardial membranes. There's a parietal, or just two layers of that. The ones around the lung are called the pleural membranes. The ones in the abdominal cavity are called the peritoneal membranes. Peritoneum. The two layers are referred to as there's a parietal layer. That's always going to be the one lining the cavity. There's a parietal peritoneum lining the inside of your abdominal wall. There's a parietal pericardium. There's a parietal pleura. Those are always the membranes on the outside, and they're usually attached to the outer layers. Like the parietal peritoneum, when you cut open your calf, you'll see it. It's just stuck to the inside of the body wall. It's fused to it. Then where it folds back on itself, the layer that covers the individual organs is the visceral layer. So this would be the visceral peritoneum. And that is the peritoneal cavity is what's actually filled with serous fluid. The size of mine is a bit exaggerated. This is a naming they use for all of them. They just change the name. Peritoneal membranes, abdominal cavity. Parietal and visceral peritoneum. Around the heart, we'll see it's parietal and visceral pericardium. Lungs, it's parietal and visceral pleura. So the name changes, but you can call them all serous membranes, and they all have this lubricating function. There may be additional functions added onto them, but they all produce serous fluid, allows things to slide around real easy. And I should also mention some of the structures we'll look at occasionally, actually starting today, are made up of these serous membranes where they have taken additional roles. Like for example, this right here, where that membrane folds back on itself, it is a serous membrane, but that forms mesenteries we'll see hold the intestine in place. Today on the uterus, we'll look at the broad ligament. That's actually what the broad ligament is. It's where the membranes cover the abdominal cavity, fold back on themselves, then cover the uterus. It forms a sheet that actually helps hold everything in place. It's reinforced with connective tissue. So these will come up a bit because they have a couple of interesting functions. Like I said, they're going to do much with the male reproductive system. I'm just going to mention them, but I wanted to get through them before we start. And a couple of things to mention. When you give injections, I don't work on humans, they don't let me. But if you do, there's several different ways to give injections, right? You got, what is it, subcutaneous, just under the skin. Then you got intramuscular, where they kind of jab it in there. Then you got intravenous, where they put them actually in the blood vessels. And you ever heard of intraperitoneal injections? Not as common, wouldn't want to give them, wouldn't want to receive them. But there are intraperitoneal injections. That's where you take the syringe and you're literally, right in there. You guys probably all feel. You remember when you were little, you talk about rabid dog, you got bitten by a rabid dog, you had to get like 15 shots in the stomach. You ever hear that sort of, I don't know, wives' tale or urban legend? Literally. Would you give someone a shot in the stomach? No, it's easier to do it here, less painful, goes to the same spot. But what they used to do with post-exposure rabies shots, it wasn't in the stomach, you're a kid, this is your stomach, it doesn't matter where you are. But they would give intraperitoneal injections. If you get a medication or a drug in here, this is all covered in blood vessels. It absorbs really, really fast. So they used to give intraperitoneal injections. They still do for some things, not the post-exposure rabies shots anymore, it's just intramuscular. But that's where they will apply some medications. If you really need it delivered quickly, that seems to work well. Also dialysis. You know what dialysis is, right? Your kidneys shut down completely. They actually pull your blood out, run it through a machine, it pumps it through dialysis tube. They have a fluid on the outside, the dialysate, that allows waste to diffuse out, that sort of thing. If you saw some kidney function, they will conduct some dialysis in here. Rather than pumping your blood through a kidney machine, they can actually put dialysis fluid in the peritoneal cavity. And the same thing, because it's good blood supply. It's just like dialysis. Toxins will diffuse out, then they can pull that fluid out, get rid of it, that sort of thing. Also, acute appendicitis. That's affecting this. We'll look at it, towards the end of the semester. But when your appendix ruptures, you're basically spewing fecal matter all over this thing. So there's a really severe acute infection. It might be peritonitis is really what appendicitis is. You spew fecal matter and all the bacteria all over those serous membrane for a nasty and without treatment lethal infection. All right, that is the serous membranes. And I'll mention, when I had that goat testicle out the other day, remember I had the spermatocord and I had that one bag thing flapping out here? That was actually what they call a tunica vaginalis, which was the parietal peritoneum. As it gets pulled down to the testes, it forms this loose bag around the testes. It's not on your lab terms. I mean, you can see it on a goat testicle easy, but that's separate from the tunica albuginea they use. But that's what I was holding up. It's part of that peritoneal membrane that basically travels down to the testes and gets deposited in the scrotum because it was up in the abdominal cavity. We'll call it quits there. Lab today. You got your lab sheets. I think I gave you last time, right? Female reproductive on one side, development. I'll go over a few things on the female that are, weird on the models. I will go over development a bit. We haven't done anything with that. It's just a couple of models. So I'll go over all the development terms when we get up there. No lab quiz today, but I'll probably give you one next week. It's the most hard takes me. Just so you don't get too hard. Keep down the street. All right, I'll see you up in the lab in a minute. These